Oral cancer is one of the most common malignancies worldwide and has a poor 5-year survival rate, which unfortunately hasn't changed in the last few decades. With recent advances in screening for this disease, there has been a tremendous increase in the number of early-stage oral lesions that can now be detected. However, only a small portion of these lesions actually progress into the more severe disease stages. And so, now more than ever, molecular signatures are needed to help clinicians isolate these lesions from the bulk of all the non-threatening ones and then assign proper treatment strategies. I'm especially interested in investigating the roles of small regulatory molecules called microRNAs in the development of oral cancer. As you can see in the slide, these molecules control the expression of various genes that either inhibit or promote cancer progression. So, when certain microRNAs are present at abnormal levels in the cell, their target genes are affected as well, and this can potentially lead to cancer. Through my research, I want to determine exactly which microRNAs are deregulated in the earliest stages of oral cancer, and then select the ones that can be used for clinical purposes. From each of the patients in my study, both normal and diseased tissue is collected at the same time from a single region in the oral cavity. I then use a molecular biology technique called quantitative real-time PCR to measure the levels of microRNAs in these biopsies. And so far, I've examined 9 tissue samples from 3 patient cases for the expression of 742 human microRNAs. Across this pool of samples, I've detected microRNAs whose levels were consistently altered in diseased tissue compared to normal tissue. Of course, to confirm these results, I'm currently adding additional patient cases to my study, and after narrowing down the list of potential candidate microRNAs, I will compare their expression patterns with patient clinical characteristics such as survival rates. And so, this type of analysis will help identify any potential associations between these clinical features. Ultimately, through the study, I will discover molecular markers that can help predict the aggressiveness of early-stage oral lesions and indicate when urgent intervention is required. In addition, these newly discovered molecules could potentially function as future therapeutic targets and hopefully help reduce the incidence of oral cancer deaths.